about videotaping your seminar? Okay, that's fine.
Yeah.
Miss, we could move a chair out if you wanted to roll your phone. Okay. Just going to wait for another minute or two for Steve. The normal Georgia Tech schedule is something that says 11 o'clock, really starts at 11:05. So
Should wind the clocks forward.
there's plenty of room up front. There's Craig. Craig, save a seat for you up front. Well, I think we'll go ahead and get started. There are still a few people coming into the room, but it's a real pleasure to have Steve Dalton with us today. Steve is a professor at the University of Georgia. He is a Georgia Research Alliance eminent scholar. Also, I guess, a Georgia Cancer Coalition eminent scholar. That's good. You've been able to tap both sides. But originally from Australia, I guess you'd say his formal education is Australia, with his Ph.D. from Adelaide University, and then went off to Vienna to do a postdoc, and following Vienna went to the Imperial Cancer Research Fund situation office there in London, and had a variety of stops, but was considered a major recruit when he was brought to Georgia as an eminent scholar. Clearly, he has been highly active in the stem cell area, and in fact, our seminar today is in part sponsored by Serologicals, which has provided us some money to bring in stem cell speakers. There are seats up front. Don't hesitate. Now is the time. So, without any further ado, Steve, welcome to Atlanta, welcome to Georgia Tech. With the exception of football Saturdays, we're always welcome to come as a representative of UGA.
Thanks, Bob. It's a great pleasure to be here. I've looked forward for a long time now to visit the Institute and to meet with you all.
I meant to comment, I wasn't sure if you were going to talk about surfing in Australia or...
No, I won't be talking about surfing in Australia today, but I can talk about it at the end of my talk if you like. I'm more than happy to. What I am going to talk about, though, is the work we've been doing over the last 5 or so years. My lab's broken up into two. We work half on human embryonic stem cells and trying to learn how to differentiate them into specific cell types that can be used for cell therapy. And the other half of the lab is devoted to looking at the biology of early embryonic development in the mouse. There is late pre-implantation and early post-implantation development, trying to look at how cells in the embryo commit to become one of the 3 germ layers. And of course, to do biochemistry and molecular biology, it's important to have in vitro models. And the in vitro model we use to model early developmental events are embryonic stem cells and their differentiated derivatives. And what I want to talk to you today is what I consider to be some of the most remarkable properties about embryonic stem cells. We can talk about the link between mouse ES cells and human ES cells at the end if you're interested. But I want to talk specifically about the mouse embryonic stem cell system, but of course there will be overlap between human as well. Now for me, the two remarkable things about embryonic stem cells are that they have this unlimited proliferative potential. They have the capacity, if grown under the appropriate conditions, to divide indefinitely. So they're immortal, and they express things like telomerase. If you look at the profile of oncogenes and tumour suppressor genes that are expressed in embryonic stem cells, they very much look like a tumour cell. Embryonic stem cells are in fact tumorigenic when placed into a heterologous environment. If injected into an immunocompromised mouse, they will form tumours, and as cells differentiate, they lose this tumorigenic potential. So I'm going to talk about cell cycle control mechanisms, and we'll be talking about molecular mechanisms of cell proliferation and how cell division in embryonic stem cells is fundamentally different to all other cell types. And then I'll talk about maintenance of pluripotency. Now, this is the other thing that's important about embryonic stem cells. It's not that they can just divide indefinitely, so they're immortal. They have the capacity to differentiate into practically any cell type in the body. So they have this amazing developmental plasticity. So embryonic stem cells can divide indefinitely while at the same time retaining their potential to differentiate into virtually any cell type. And those two properties together are known as self-renewal. I'm going to talk today about the molecular regulation of self-renewal, how a stem cell divides indefinitely, and how it retains its developmental potential. Now, there are two broad classes of stem cells which you'll be aware of course. The adult stem cells, which are found in organs in the body, in the adult. And these cells typically divide through asymmetric cell divisions at mitosis, which generate two daughter cells, one which retains stem cell identity, and the other one which doesn't retain stem cell identity, which will then differentiate to generate more specialist cell types. So we have asymmetric cell division in adult stem cells. Embryonic stem cells divide to generate two equivalent daughter cells. So we have a symmetric mitosis. Both of the daughter cells at mitosis retain stem cell identity. So that's what we're dealing with when we're talking about cell division of embryonic stem cells. Now, embryonic stem cells have this amazing capacity to differentiate into basically any cell type in the body. I mentioned previously that the stem cells are also quite tumorigenic. And as part of this differentiation program, they lose tumorigenicity. And we were very interested in understanding what the link was between self-renewal, immortalization of the stem cells, and tumorigenicity. And one of the things we focused on initially was cell cycle control mechanisms, because many of the regulators of cell division cycle controls are in fact oncogenes and tumour suppressor genes. So it could be related to their tumorigenic properties. Now, what got us interested in this general question in a developmental context was that the pluripotent cells in the embryo divide at very unusually rapid rates. So this is a summary of work done around about 25 years ago now by Snow. And basically it summarises the number of pluripotent cells in a specific stage of embryonic development. Now this is a picture of a mouse embryo, and what I'm going to be talking about are just these purple cells here. These are the pluripotent cells which will generate the embryo proper. All of the other tissues and cell types that we have here are extraembryonic and are really supportive tissues for the embryo. So the purple cells are pluripotent cells. Here we have days postcoitum, so days after fertilization, and here we have cell number of the pluripotent cells in the embryo. So these are the cells which are the embryonic epiblast. And here at the bottom we have average cell cycle time, which really is deduced by just counting the number of cells, pluripotent cells in the embryo in staged embryos. So by counting the number of cells at different stages, you can deduce the average cell cycle time. And what's quite remarkable about this stage of development, around about day 6.5, is that the average cell cycle time is in the order of 4.5 hours. Some of the cells are dividing faster, some are dividing slower. But this is a remarkable rate of cell proliferation for a mammalian cell. This won't impress Drosophila and Xenopus developmental biologists because they undergo their own unique rapid mode of cell division in the early cleavage division and syncytial cycles. But for a mammalian cell, this is a very rapid rate of cell division. So we go from a brisk rate of cell division to an incredibly rapid rate of cell division. And as cells differentiate in gastrulation, and I'll talk about gastrulation in a minute, the cell division cycle slows down again. So we have this proliferative burst followed by slowing down of cell proliferation when the pluripotent cells in the embryo differentiate. Now, differentiation of the pluripotent cells in the embryo occurs during a process known as gastrulation. And gastrulation is the point in development that occurs around about day 6.5 to day 7, where the pluripotent cells, which previously are uncommitted to form any of the 3 germ layers, become committed to form either ectoderm, endoderm or mesoderm. So that's the process known as gastrulation. It's a key event in development because the pluripotent cells become committed to become a specified cell type. Now, coming back to our model systems in vitro, if you actually look at the number of cells in the embryo, the pluripotent cells in the embryo at this stage of development, we're looking in the order of several hundred. You simply can't do biochemistry on this number of cells. What we can do, however, is surgically dissect out the pluripotent cells from the embryo. So at this stage here, we're post-implantation. So you can surgically dissect out the embryo from the decidua. Then you can dissect away the extraembryonic tissues and then peel away the pluripotent cells from the extraembryonic endoderm and get pure pluripotent cell populations. But on a per embryo basis, you're only going to have several hundred. So you can't do serious biochemistry and molecular biology on this sort of tissue because it's just the amount of materials is not large enough. So we have to use in vitro systems, and the in vitro systems we use are embryonic stem cells, which are the in vitro equivalent of the inner cell mass stage embryo, the blastocyst embryo. And we use another cell type known as EPL cells, which are a slightly later stage of pluripotent development, which are equivalent to the primitive ectoderm. And if you actually look at one of the things we're interested in doing was trying to understand why these cells divide so rapidly. And it's not just in the embryo the pluripotent cells divide rapidly. The pluripotent cells in vitro divide rapidly as well. Embryonic stem cells divide in the order of 10 hours. The primitive ectoderm that we can make, which is still pluripotent, divide in the order of 8 hours. This is very rapid cell division. And then during their differentiation, these pluripotent cells cycle at a much slower rate. Now, this is some propylium iodide stained profiles of embryonic stem cells and these EPL cells, which are still a pluripotent cell population. And what you'll notice, if you've ever seen a PI profile before, is that we have a very high proportion of cells in S phase and a very low percentage of cells in the G1 phase of the cell cycle. Now, in most cell types, cells spend most of their time in G1. G1 is a very important part of the cell cycle because that's when cell volume increases, cell growth increases, and the cells respond to mitogenic signals which cue them to either undergo another round of replication or to enter quiescence. So the G1 phase is normally very important and much longer than most of the other phases. The pluripotent cells, both in vitro and in the embryo, have a very unusual cell cycle structure. And part of the reason for this is that they truncate their G1 phase and basically come out of mitosis and then go straight into S phase. And this all in all accounts for rapid rates of cell division. And then you can differentiate pluripotent cells into embryonic bodies by growing them in suspensions in the absence of maintenance factors such as LIF. And then the cell division rates will slow down, and then the cell cycle completely remodels and starts to look now more like a HeLa cell or an NIH3T3 or a mass embryo fibroblast. We have this very high percentage of cells in the G1 phase. Of course, the cell division cycle, we're interested in understanding what the molecular basis was behind this very rapid mode of cell division in the pluripotent cells and how that changes as pluripotent cells differentiate into one of the 3 germ layers. And the molecular basis behind cell division is that cell division cycle phase transitions are driven by a family of protein kinases known as cyclin-dependent protein kinases, which phosphorylate proteins required for the different cell cycle transitions. So for each specific cell cycle phase, there are cyclin-dependent kinases which are activated at precise points of the cell cycle, and their job is to phosphorylate proteins required for things like DNA replication and mitosis. Okay, so we have different flavours of cyclin-dependent kinases required for the different cell cycle transitions. And a cyclin-dependent kinase consists of the catalytic subunit, a CDK, and a regulatory subunit known as the cyclin. And it's only when the cyclin binds the catalytic CDK that the complex becomes active. The important thing is that cyclin protein levels are in fact cell cycle regulated, so that cyclin proteins are periodically synthesized at specific points of the cell cycle and then degraded at specific points of the cell cycle, so that cyclin levels are in fact cell cycle regulated, thus allowing for cell cycle regulation of the kinase activity. Now, if we look at the cell cycle structure, this is now, we've taken 20 day 6.25 day embryos, and this is an embryo that's been dissected out of a decidua. It's a whole mount in situ, and we're staining the embryo with an anti-OC4 antisense RNA probe. So this is lighting up the pluripotent cells in 6.25 day embryos. We took a total of 20 embryos, surgically dissected out these pluripotent cells here, made single cell suspensions and stained them with propydium iodide, and we get this sort of cell cycle structure, which is very similar to what we see the pluripotent cells in vitro. So the cell cycle structure of pluripotent cells in the embryo is very similar to the in vitro pluripotent cells that we work with. And this is just an example of two cell lines, NIH3T3 fibroblasts and mouse embryo fibroblasts, which have a totally different type of cell cycle structure. Now, what's the basis underpinning this rapid rate of cell division and unusual cell cycle structure? Now, we know that cyclin-dependent kinases are the orchestrators of cell division, so we asked, well, could it be that cyclin-dependent kinases are very active in pluripotent cells and they drive rapid rates of cell proliferation? So what we did was to essentially perform kinase assays on embryonic stem cells. So we made whole cell lysates and then immunoprecipitated either the catalytic subunits of the kinase, CDK, or the cyclin regulatory subunits. And then to the immunoprecipitates, we then added histone H1 as an in vitro substrate and P32, and then rad the kinase assay out on a gel and then put it down on film. And this represents P32 labelled histone H1, which has been phosphorylated by the cyclin-dependent kinase in the ES cell lysate. Now, I just want to draw your attention to the levels, relative levels of cyclin-dependent kinase activities in pluripotent cells compared to differentiated cell lines. We're looking at least tenfold greater than any other cell line that's been characterised previously. So pluripotent cells, embryonic stem cells, have very high levels of cyclin-dependent kinase activities, and in a number of cases now, it's been shown that upregulation of cyclin-dependent kinase activity in tumour cell lines is responsible for driving rapid rates of cell proliferation. So we think that the molecular basis behind rapid rates of cell division and this very unusual cell cycle structure is the very high levels of cyclin-dependent kinase activities in the pluripotent cells. And I'm not going to talk much about the cell cycle work today because that's published, but just to summarise what we know about cell cycle control in embryonic stem cells. They divide at very rapid rates. They have short gap phases. CDK activity is unusually high. I just showed you that on the previous slide. And importantly, the cyclin-dependent kinase activities that you have in embryonic stem cells are not cell cycle regulated. One of the principal features of cell cycle regulation is that you switch off cyclin-dependent kinases at specific points of the cell cycle and then switch on other cyclin-dependent kinases. You have this oscillation where kinases are switched on and switched off, thus defining the different cell cycle transitions. In embryonic stem cells, there's no cell cycle regulation of these kinase activities, with the exception of the mitotic kinase CDK1 cyclin B. So these cells are very unusual, and in fact, if you were going to look at the molecular profile in terms of their proliferation, you would say they're incredibly tumorigenic based on the activity of the proliferative machinery. Many of the tumour suppressor functions that normally exist in normal cells, which restrain cell division, are in fact not functional in embryonic stem cells. The retinoblastoma tumour suppressor family are inactive in embryonic stem cells because they're held in a biochemically inactive state by phosphorylation through CDK activity. So RB tumour suppressor proteins can be regulated by cyclin-dependent kinases. When they're phosphorylated, the tumour suppressor protein family is switched off. In the embryonic stem cells, because we have such high levels of CDK activity that's active throughout the cell cycle, the RB family are held in a biochemically inactive state. And we also have a distinct absence of inhibitors of cyclin-dependent kinases, which also are implicated in tumour progression. So basically, the whole proliferative machinery of embryonic stem cells is wired to divide in more or less an uncontrolled, rapid manner. But what happens during differentiation? Some very significant changes. These cells go from looking, at the molecular level at least, from looking like a tumorigenic cell, which divide rapidly in an uncontrolled manner, to a cell that now starts to respond to its environment and has very strictly controlled cell cycle regulatory mechanisms. And you can differentiate embryonic stem cells as embryonic bodies by growing them in suspension in the absence of lift, for example, And then what happens to these CDK activities is they start to collapse and decrease during differentiation. So both the kinase activity and the protein levels of the cyclins decrease during differentiation. And this occurs at a point around about where differentiation starts to occur. So this is a northern blot just showing you a number of RNA markers which come up during differentiation. Brachyury is a marker for nascent mesoderm formation. This is indicative that differentiation is starting to occur at day 3, and this coincides with the downregulation of these cyclin-dependent kinase activities. So the loss of these precocious CDK activities correlates with the activation of markers which correlate with specific differentiation programs. So during differentiation, the cell cycle length increases. You have a restructuring of the cell cycle, so where you go from a cell cycle that lacks G1 phase control, you go to a cell cycle which has a long G1 phase control. You have the establishment of cell cycle regulated CDK activities, and importantly, we can talk about this later, you start to have signaling from the outside of the cell controlling the cell cycle machinery, so the cell now is more responsive to its environment. So this sort of summarises the proliferative control in ES cells. We have these very high levels of constitutively active CDK activities, which are very similar to control of CDK activities in tumour cells. And then as part of the differentiation program, which is part of the natural differentiation program that occurs in the embryo as well, you get a decrease in CDK activities, and then they start to become cell cycle regulated, just as it's all written about in undergraduate textbooks. Okay? So we have a very unusual situation in the embryonic stem cells which differs markedly from the molecular control of cell proliferation in differentiated cells. So I think that goes a long way to explain that the tumorigenic properties of embryonic stem cells, the biochemical properties, the biochemical mechanisms underpinning their cell division is much more like a tumour cell than a normal cell. But what happens during differentiation is that these tumorigenic features are lost, and then the cell cycle starts to become cell cycle regulated like other cell types. So I want to talk now more about molecular mechanisms of stem cell maintenance. What are the mechanisms which maintain a stem cell to allow it to divide indefinitely and to retain its full developmental potential? And you'll know in mass embryonic stem cells, the way you grow cells is typically in the presence of IL-6 family member cytokines such as LIF, or leukemia inhibitory factor, which binds to a heterodimeric receptor, GP130 LIF receptor. This then recruits latent inactive STAT3 transcription factor, which is in the cytoplasm, to the nucleus, where it becomes phosphorylated by receptor tyrosine kinase activities. Now, following phosphorylation at the receptor, STAT3 then translocates to the nucleus, and the idea is that STAT3 then activates genes required for stem cell maintenance. Now, the genetic program required for stem cell maintenance and stem cell self-renewal has not been defined previously. It's a big black box. We know that LIF is involved in docking to the receptor. It activates STAT3, but we don't know what happens after this step. An interesting experiment is you can actually grow cells in the absence of LIF by ectopically expressing an active form of STAT3. So we know that active STAT3 under most conditions is sufficient to maintain self-renewal. So that everything downstream of STAT3 is of course going to be crucial to understanding the self-renewal mechanisms. So we answered this question by trying to ask, well, what are the targets of STAT3? How is STAT3 working on embryonic stem cells to maintain self-renewal? Now, just sidestepping for a minute, there are a number of transcription factors which could be perceived to be downstream targets of STAT3 at the transcriptional level. OCT4, which is a paidomain transcription factor expressed not only in the pluripotent cells in the embryo but also the germline. It actually is starting to occur in many adult stem cell populations as well. NANOG, a homeodomain transcription factor described last year by two groups, SOX2, UTF1 and FOXD3. These are all transcription factors which have some important role in development of the pluripotent cells in the embryo. But I just want to say these are not the targets of STAT3. If you express any one of these ectopically, they will not maintain embryonic stem cells in the way that STAT3 will. Another important point that I just want to mention is that there's an alternative way of maintaining human and mouse embryonic stem cells that's been reported, and I'm not totally convinced that this story holds together in its entirety, but let me just mention this much. It's been reported by Brivenloo's group that Wnt signaling can maintain embryonic stem cells in a self-renewing state. So this is both mouse and human embryonic stem cells. And more importantly, that the Wnt signaling acts to suppress GSK3 activity. And it's argued that suppression of GSK3 activity is important for promotion of the self-renewing state. So you need low GSK3 activity in order to be a stem cell. Okay, so just remember that, because I'll be coming back to that later in terms of trying to provide a mechanism to explain this. So we have two potential pathways that can maintain mouse embryonic stem cells. We have the LIF-STAT3 pathway, which activates self-renewal genes, a genetic program of transcription. But this can be replaced. We can take away LIF and then add Wnt or Wnt conditioned media. And Wnt works through beta-catenin TCF-LEF and activates target genes, presumably, which the same target genes that maybe LIF and STAT3 are acting through. So one of the questions we asked was, how do separate signalling pathways independently promote self-renewal? These are very different signalling pathways, but they can substitute for each other. You can take away Wnt and give the cells LIF, you'll maintain self-renewal, and vice versa. So we reasoned that perhaps these two signalling pathways, although very different, were focusing in on the same set of target genes and promoting self-renewal in that way. I'll come back to that in a minute. There are several other levels of complexity when you consider self-renewal of embryonic stem cells. What a stem cell basically is, the way it's starting to look, is that a self-renewing stem cell is a cell that's blocked in its ability to differentiate into the three germ layers. And in order to maintain a stem cell, you need to provide multiple lineage-specific differentiation blocks. And there's evidence now that factors in serum, for example, in conjunction with LIF, are required for self-renewal of mouse embryonic stem cells. And the way this works is that BMPs, which is the key component of serum, suppress neural differentiation, and other factors in the culture conditions suppress either endoderm or mesoderm differentiation. So these are the three main germline specific differentiation pathways that an ES cell can go down. In order to maintain self-renewal and to maintain pluripotency, you need to block differentiation down these pathways. Okay, now coming back to the LIF-STAT3 story. We were very interested, I've talked already about the cell cycle, and we were very interested in sort of trying to establish links between cell cycle control mechanisms. I've mentioned the very unusual mode of cell cycle control in the embryonic stem cells. So we were interested in identifying links between these cell cycle control mechanisms and stem cell self-renewal. So for example, could the unusual mode of cycle-dependent kinase activity actually be intrinsically involved in self-renewal mechanisms? Another important point is we know that LIF and Wnt appear to substitute for each other in their role in promoting self-renewal. So we reason that they could be working by targeted common downstream effectors. So the hypothesis we came up with, trying to pull in cell cycle control and LIF and Wnt together with this idea that there's a common downstream target for the LIF and Wnt pathway, is that MYC family members could be involved in self-renewal, because MYC, c-MYC in particular, is a known target of STAT3 dependent and Wnt dependent signalling pathways in other cell types. So there are precedents to link c-MYC regulation into LIF and Wnt dependent signalling. And c-MYC is a helix loop helix, basic helix loop helix transcription factor, which dimerizes with its obligatory binding partner MAX, goes to the nucleus and can either activate or repress target genes in the nucleus. And it has very many pleiotropic functions. MYC has been the most elusive target gene in terms of trying to identify cellular functions for, because it regulates so many different genes. Chromatin immunoprecipitation assays have shown that MYC is bound to around about 10% of all active genes in the cell. So trying to say that MYC is involved in proliferation or apoptosis or differentiation is a bit naive. What I think MYC is, it's a chromatin remodeling factor which binds to many promoters and recruits other components of the chromatin remodeling machinery, and then allows those genes to respond to signals that are coming in from outside of the cell. So in different cell types, MYC will regulate very different repertoires of genes. So you can't predict what effect MYC overexpression or MYC underexpression will have on a given cell type. It will vary depending on the cell type you're dealing with. These are sort of a short list of the different genes it's been involved with, but MYC has its hand in a lot of pies. Differentiation, apoptosis, tumorigenesis, cell adhesion. So by just saying what would you expect MYC to be doing in an embryonic stem cell is very difficult, because it has so many defined roles. And of course, there are over 20 microarray papers, at least, probably more now, that have tried to identify MYC target genes, and no clear consensus in terms of target genes have been established through their extensive transcriptional profiling studies. So the hypothesis was that maybe STAT3 and Wnt are feeding in and promoting self-renewal through a MYC-dependent mechanism, maybe MYC's involved, because we know they're both downstream. MYC is downstream of both STAT3 and LIF. So in order for MYC to be a regulator of self-renewal, we would expect that MYC would be expressed in embryonic stem cells, and that perhaps its transcriptional profile will become down-regulated during differentiation. So this is some RT-PCR analysis, just looking at mRNA levels in embryonic stem cells and then in differentiating embryoid bodies. And we start off with a small panel of differentiation markers. REX1 is a classic ES cell marker, which is also expressed in the inner cell mass in the embryo. OCT4 is more representative of the broad spectrum of pluripotent cells, not just the inner cell mass ES cell group. FGF5 is a marker for primitive ectoderm, and brachyury is a marker for nascent mesoderm, and in fact mesendoderm. And then so here we can sort of stage where we are in differentiation. Here's c-MYC. c-MYC is high in embryonic stem cells and then gets downregulated rapidly during differentiation. You have low levels there throughout, but it's very high in embryonic stem cells and then downregulated. And remember, when these cells are differentiating, they're not going terminal. They're not going post-mitotic. These cells divide and form specialist cell types up for several weeks. So this is not just the fact that the cells are exiting the cell cycle, the cells are still dividing. So the first part of the idea was, yes, MYC is there in embryonic stem cells and it gets down-regulated during differentiation, which would perhaps point to a role for MYC in the stem cell state. This is an experiment showing that MYC is directly regulated by LIF-STAT3 signaling. So what we've done is to grow embryonic stem cells in the absence of LIF. So you go from high c-MYC mRNA, you take away LIF, it goes down, and then you give LIF back, and c-MYC mRNA levels go back up. So c-MYC transcript levels are responsive to LIF-dependent signaling. Now, the next question is c-MYC a direct transcriptional target of STAT3 in vivo? So we need to show, is STAT3 actually activating the c-MYC gene in an embryonic stem cell? And we need to show this in order to validate the hypothesis. So what we've done is to perform in vivo assays, chromatin immunoprecipitation assays, where we grow the cells and then add a cross-linking agent to the cell culture, which cross-links transcription factors to the DNA. So you cross-link, and then you can basically harvest the cells, shear the chromatin with a sonicator up into fragments of around about 1 kb in length, and then immunoprecipitate the STAT3 protein from the cell lysate. And if the STAT3 is cross-linked to a promoter in vivo, it will pull down the chromatin fragment with it, the DNA promoter fragment with it. And then you can simply go into the immunoprecipitate and then use primers specific for the c-MYC promoter to ask, has the c-MYC promoter been precipitated with the STAT3 transcription factor? And this will be evidence that c-MYC is bound to the c-MYC promoter in vivo. So what you see in embryonic stem cells, STAT3 is bound. When you take away LIF and differentiate the cells, this binding of STAT3 to the c-MYC promoter decreases. And this is totally consistent with the inactivation of STAT3 following LIF withdrawal. So when you take away LIF, cells differentiate because STAT3 becomes inactive. And so we have a nice correlation now between STAT3 and the c-MYC gene. The next question is, well, can STAT3 actually transactivate the c-MYC gene in vivo? And what we've done there is to make a cell line where we stably express a fusion protein, where we express STAT3 fused to the steroid binding domain of the estrogen receptor. And what this is, it's basically an inducible system whereby, in the absence of added estrogen analog 4-hydroxytamoxifen, we can express the estrogen receptor STAT3 fusion protein in an inactive form that essentially accumulates in the cytoplasm. However, when we add 4-hydroxytamoxifen, it binds to the estrogen receptor, and then the STAT3 ER fusion translocates to the nucleus and activates genes that are presumably involved in self-renewal. So it's an inducible system whereby we relocalize the latent form of the protein in the cytoplasm into the nucleus. And this works very nicely for transcription factors. So here's the experiment. Cells grown in the presence of LIF, of course, have high levels of c-MYC and high levels of OCT4. We take away LIF for 36 hours and c-MYC transcription goes down. And then to this pool of cells, we can then either add estrogen analog 4-hydroxytamoxifen, which activates the STAT3 protein, or we can add lift back. And in both cases, you get rescue of c-Myc transcription. So what this shows us is the c-Myc gene, it can be directly regulated by STAT3. So we have 3 lines of evidence to link the lift STAT3 signalling pathway to c-Myc. First of all, c-Myc is responsive to lift signalling in ES cells. Secondly, STAT3 binds the c-Myc promoter. And thirdly, STAT3 transactivates the c-Myc gene in vivo. Okay. So c-Myc is a direct target of lift-dependent STAT3 signaling in ES cells. So far, this is consistent with the hypothesis that Myc is part of the self-renewal mechanism being downstream of STAT3. Now, I just want to talk for a few minutes about Myc and Myc regulation in embryonic stem cells. If you actually look at the Myc so far I've talked about Myc mRNA levels, and I want to talk about Myc protein because there are 2 levels of control which control Myc. It's through a transcriptional mechanism which involves STAT3 and by controlling protein levels. So this is a Western blot looking at c-Myc protein levels in ES cells, and then during differentiation you see that the levels of c-Myc collapse following lift withdrawal as ES cells differentiate. Now, we noticed in several experiments that the c-Myc protein was actually disappearing before c-Myc messenger RNA was going down, which indicated there were some post-transcriptional levels of control involved in controlling Myc levels in ES cells. And one of the best characterized mechanisms which regulates c-Myc protein levels in the cell is through protein stability. And protein stability of c-Myc is often controlled through its phosphorylation on a residue 3-ene-58. So when 3-ene-58 is phosphorylated, c-Myc becomes ubiquitinated and then gets rapidly degraded. In the unphosphorylated state on T58, c-Myc is stable. So we look to see whether phosphorylation on 3-ene-58 correlated with the downregulation of c-Myc during differentiation. And you'll see straight away that in embryonic stem cells, c-Myc is not phosphorylated on 3-ene-58, which would indicate the protein is not going to be targeted for destruction. But following lift withdrawal, you start to get T58 phosphorylation, which is coincident with the downregulation of c-Myc. Now, the kinase that phosphorylates 3-ene-58 has been characterized in quite a amount of detail, and it's GSK3 beta. And remembering back to earlier on, I said that there have been published reports now saying if you use inhibitors of GSK3 beta activity, it can promote a self-renewal of both mouse and human embryonic stem cells. So what I'm trying to lead you towards is a possible mechanism whereby GSK3 activity is important for self-renewal through controlling c-Myc. So the question would be, well, if GSK3 beta is important in controlling c-Myc protein stability, then you would expect that GSK3 beta would be inactive in embryonic stem cells and then perhaps will become activated very early during differentiation. So we assessed GSK3 beta activity using a kinase assay whereby we made whole cell lysates from embryonic stem cells and then differentiated cells, and then immunoprecipitated GSK3 from whole cell lysates, and then used myelin basic protein as an in vitro substrate to evaluate GSK3 beta activity. And this is a radioactive assay where we have P32 labelled myelin basic protein. And what we see in embryonic stem cells is no GSK3 beta activity, but within 1 day of withdrawal of LIF, as the cells start to differentiate, GSK3 beta activity is elevated around about 15-fold, which ties in nicely with the kinetics of phosphorylation on T58. Importantly, the mutation of T58 pops up in very many cases where Myc functions as an oncogene. In Burkitt's lymphoma, for example, mutation of c-Myc at residue T58 contributes to the transforming oncogenic capacity of c-Myc, because essentially what the mutation does is to stabilise c-Myc and elevate c-Myc levels in the tumour cell. So as opposed to having gene duplication or chromosomal translocations, a point mutation of c-Myc can enhance c-Myc protein levels by increasing its stability by the same residue that we see here being crucial for regulation of c-Myc levels in embryonic stem cells. And this is just pointing out the key residue here, T58, lies in the transactivation domain of c-Myc. Now, what I haven't talked to you about so far is c-Myc actually really a stable protein in embryonic stem cells? The implication is that it would be, because it's not phosphorylated on T58. And we have some anecdotal evidence to suggest that because the protein levels go down before transcription, then there is a differential protein stability following lift withdrawal. So this is actually an experiment addressing the question of, well, how stable is c-Myc in embryonic stem cells? Now, the most stable form of c-Myc that's been reported in the literature is in cell lines with Burkitt's lymphoma. And that's in the order of 90 minutes. So Myc is generally a very unstable protein. It has a half-life in the order of 15 minutes in most cell types. And that's shown here in NIH3T3 cells, for example. This is a cyclohexamide chase experiment where we've made extracts in NIH3T3 cells treated with cyclohexamide for various amounts of time, and then we're looking at the disappearance of the pre-existing c-Myc. So we're blocking new synthesis and then just looking how quickly the existing protein disappears. And you can see that the half-life of c-Myc in NIH3T3 cells is very short, less than 15 minutes, and this is typical of basically every cell type that's been non-tumour cell line that's been characterised in the literature. If you look at the half-life of c-Myc in embryonic stem cells, so we're comparing the authentic c-Myc, the endogenous c-Myc, and this is a transgene expressing a MYC-ER fusion. The stability of MYC in embryonic stem cells is extraordinary. It actually exceeds what you see in Burkitt's lymphoma. So it's at least 90 minutes and probably 120 minutes. So we have a very unusually stable form of c-Myc in embryonic stem cells, and we think part of the basis for this is that GSK3 activity is suppressed. So you don't get phosphorylation at T58, so MYC doesn't get turned over. This is another experiment just showing now this is a MYC-ER fusion. The MYC-ER fusion expressed from a transgene behaves indistinguishably from the endogenous authentic c-Myc. You take away LIF, and by day 2, the protein's gone. So you have this very, very tight regulation of c-Myc protein in embryonic stem cells, and that as part of differentiation, c-Myc protein levels collapse. If you actually mutate the T58 residue and do the same experiment and allow the cells to differentiate, the MYC protein persists. So this demonstrates that it is the T58 residue that's crucial for MYC stability in ES cells, and again implies a role for GSK3 beta in controlling MYC stability. And again, this just reinforces even more the significance of stability of MYC in embryonic stem cells. Here we have an experiment where we have ES cells and then embryonic bodies. So this is differentiating cells day 1, 2, 3, 4. And then what you normally see is a collapse of MYC at, say, day 2 is what you see up here. So normally at day 2, MYC levels have disappeared. But if you add proteasome inhibitor, MG132, you can rescue MYC protein levels at to day 4. So this is due to the fact you've got a little bit of residual c-MYC transcription, but because the protein is so unstable, you don't see it under normal conditions. But if you add proteasome inhibitor, you can actually see the protein through blocking its degradation. So this is good evidence that the protein goes from a stable form to a very labile form during differentiation. And we think this is because you get GSK3 beta activated very soon in differentiation. Now, to try and tie in MYC function as part of the self-renewal mechanism, it would have to be downstream of Wnt as well. So we asked whether MYC was regulated in a similar way in Wnt-maintained cells as it was in LIF-maintained cells. Of course, if it wasn't, it would be a LIF-specific type of regulation. But for it to be fundamental and core to self-renewal, we would have expected that MYC would have behaved in the same way in both self-renewing systems. So we've done the same experiments. In Wnt-maintained cells, MYC is not phosphorylated, but you take away Wnt, by day 1 it's phosphorylated, and GSK3 beta is not active in Wnt-maintained cells. You take away Wnt, cells differentiate, and GSK becomes rapidly elevated. So that the behaviour of MYC and the regulation of GSK3 is the same in Wnt-maintained cells and in LIF-maintained cells, which sort of gives extra support to the idea that MYC has some fundamental role in general aspects of stem cell self-renewal. So just to summarise what I've mentioned with regard to control of c-MYC. c-MYC is controlled at the transcriptional level by a LIF-dependent mechanism which involves activation by STAT3. So the main role of STAT3 in promoting self-renewal, I'll try and demonstrate to you, is through its transcriptional control of c-MYC. It's also important not just to have STAT3 activity, you need to suppress GSK3 beta activity in order to elevate or have c-MYC protein levels elevated, and you need to have an absence of T58 phosphorylation. Okay, so that's sort of just saying, well, MYC is expressed in the S cells, it disappears in differentiation, and here are two mechanisms by which it's regulated. One by STAT3 dependent mechanism and another by suppressing GSK3, which impacts on protein stability. But of course the burning question is, well, does c-MYC actually promote self-renewal? We've got some sort of interesting evidence to say that it correlates with self-renewal mechanisms, but does c-MYC control self-renewal? So the experiment we did was to express c-MYC from a transgene, either from a CAG promoter or an EF1 alpha promoter, hooked up to an IRES and the drug selectable marker. So we made stable cell lines expressing high levels of MYC. And then we asked the question, can elevated levels of c-MYC promote self-renewal of stem cells in the absence of LIF? So essentially you do this experiment, you make the cell lines, you take away LIF, and in the presence of enforced c-MYC transcription, can you still retain stem cell identity? And okay, so the system that we used was again this estrogen receptor fusion system, whereby we make stable cell lines expressing MYC-ER fusions, which gets expressed constitutively in the cytoplasm in an inactive form. And then by adding the steroid analog 4-hydroxytamoxifen, the tamoxifen binds to the estrogen receptor steroid binding domain, displaces heat shock proteins which are bound to the estrogen receptor, and then unmasks a nuclear localization signal, which then allows MYC to relocalize into the nucleus. And it does so in a very tightly controlled concentration-dependent manner. The more tamoxifen you add, the more MYC-ER fusion protein you can localize to the nucleus, and it's totally reversible. So you have a concentration-dependent reversible gene induction system. So we can switch MYC on and switch MYC off, and then ask, does that correlate with self-renewal? So we performed a number of preliminary assays to try and see whether expression of MYC could maintain self-renewal in the absence of LIF. So we did flow cytometry looking at cell surface markers. One of the classic for mass embryonic stem cells is SSER1, which is the carbohydrate epitope on the cell surface. Now we did this two ways. We made cell lines expressing wild-type MYC, so just normal authentic MYC, and then we expressed the mutant form of MYC, which was the stable form of MYC that shouldn't be phosphorylated by GSK3. Okay, and what we found when we expressed wild-type MYC is that, so we're taking the cells out now in the absence of LIF for up to 12 days, and then asking the question, do the cells still retain high SSEA1 staining? And essentially what happens is that these cells expressing MYC don't retain SSEA1 staining indefinitely, but they delay the loss of SSEA1 staining. In contrast, if you express the stable form of MYC, you retain SSEA1 staining indefinitely. So there's a difference between the normal MYC and the stable MYC. Normal MYC delays differentiation, but the stable MYC blocks it, or at least as judged by this marker. And then if you look at alkaline phosphatase activity, another readout for stem cell markers, in the stable form of c-MYC can maintain alkaline phosphatase levels completely in the absence of LIF up to 15 days. If we express the wild-type form of MYC, it delays differentiation. Okay, so this is consistent with what we see up here. And this is just showing that some of the transcripts associated with the stem cell state are retained in the MYC T58A fusions. We can keep on genes associated with the stem cell state by expressing stable c-MYC, but not normal c-MYC. So the logic is that you need two levels of control to maintain MYC-dependent self-renewal. You don't just need to express MYC, you need to express or have a stable form of MYC. And if you think about what was going on in the normal case in embryonic stem cells, you have STAT3 dependent upregulation of MYC transcription, which is one requirement, and secondly you need suppression of GSK3 activity, which blocks its degradation. So in the absence of LIF, if you express normal c-MYC, it'll just be degraded because GSK will get upregulated. But by expressing a mutant form of MYC that can't be phosphorylated, that then is sufficient to maintain self-renewal. So it comes back to these two levels of control required. Again, these are the MYC-ER cell lines, and these are the parental cell lines grown in the presence of LIF. Nice dome-shaped round colonies. You take away LIF in the absence of tamoxifen, both cell lines flatten out, become more heterogeneous in terms of their morphology, indicative of differentiation. If you switch MYC on in tamoxifen, the parental cells in the presence of tamoxifen differentiate as they did before, but the MYC-ER cell lines retain this nice, uniform, round, dome-shaped colony morphology. So by morphological criteria, MIC can maintain ES cell morphology. And this is just an experiment just showing that for the stem cell people that the proportion of stem cells in the bulk culture in MIC-maintained cells is equivalent to that in LIF-maintained cells. So essentially what you do is you grow cells in the presence of LIF, or you grow them in the presence of tamoxifen in the absence of LIF, and then do that for 30 days. And then take these cells out to single-cell suspension and plate them at low density and ask how many of those cells in that bulk culture can give rise to stem cell colonies. And the upshot is that the ability to maintain stem cells in the bulk cultures is indistinguishable between LIF-maintained cells and MIC-maintained cells in multiple ES cell lines. So this is not just restricted to one cell line. It's a characteristic of different cell lines that we've worked with. So do MIC-maintained cells retain pluripotency? I've shown you that if you keep elevated levels of MIC on in the absence of LIF, you can retain stem cell markers such as SSCA1, alkaline phosphatase and various transcripts. And the cells look like stem cells and they have a similar clonal plating efficiency to LIF-maintained stem cells. But are they really still truly pluripotent? The best test of the stem cell is then to test whether these cells can differentiate in a normal setting into the 3 germ layers. So we wanted to address this question by asking, well, okay, we have MIC-maintained cells. Do they still have the ability to differentiate? It could be, and you'll know this from embryonic carcinoma work, that over time some cell lines lose the ability to differentiate into certain cell types or even lose the ability to differentiate completely. We wanted to show that these cells were still capable of differentiating. So we took cells that were being grown in the absence of LIF, in the presence of tamoxifen, and then split them into 2, and then grew them either without tamoxifen or with tamoxifen, and asked the question, do you get downregulation of stem cell markers when you take away tamoxifen? And the answer is yes. You get downregulation of OCT4, upregulation of brachyury. If you keep tamoxifen on, i.e. if you keep MIC activity elevated, you don't downregulate your stem cell markers and you don't turn on brachyury. So that shows that the MIC-dependent self-renewal is reversible. If you take away tamoxifen and switch MIC off, the cells will differentiate, at least as judged by those markers. But the best assay to actually demonstrate this is to show that the cells that are being maintained with MIC in culture for extended periods of time can still differentiate into the 3 germ layers. And the best way to do that is to take those stem cells and inject them back into blastocyst stage embryos and ask, can they contribute to the 3 germ layers in chimeric animals? And that's what this experiment is. We've essentially grown MIC ER cells in the absence of LIF for at least 30 days, and this is what the cells look like. If you plate them down in the absence of LIF, they flatten out, and the absence of tamoxifen, they flatten out and differentiate, which again is consistent with them being capable of differentiating when you switch MIC off. And then what we've done, and they're alkaline phosphatase positive, and then what we've done is to take these cells which have been grown in tamoxifen, haven't seen LIF for over a month, and injected them into blastocyst stage embryos. And the trick we've done is to use a green fluorescent protein marked embryonic stem cell line expressing MIC. So these are GFP labelled ES cells from an SV129 genetic background, which has a sandy coloured coat. And these cells, which have been maintained with MIC, have been injected into a black 6 blastocyst stage embryo. And so we can easily see that the GFP MIC-maintained ES cells integrate widely into all of the tissues. As by this is around about a day 13 embryo. This is a mock injection to show this is not autofluorescence, and this is just high magnification zooms of different tissues. So we can see nice contribution to muscle, gut, heart, kidney, and coat colour. We can see chimerism there, so we've got ectodermal lineages being generated from these MIC-maintained cells. So you can see the sandy colour, coat which is generated from the injected GFP ES cells and the black host, black 6 coat colour. So we can say now that these MIC-maintained cells can be maintained as stem cells. They look like stem cells. They retain all of the cell markers of stem cells, and when injected back into a blastocyst stage embryo, they can differentiate into the 3 germ layers in a manner that's indistinguishable from LIF-maintained cells. This is you need to use the stable MIC. Okay.
So that implies it is somehow getting degraded in some other way.
No, it's not getting degraded, but when you inject it into the blastocyst, you don't have any tamoxifen there, so the levels just decline over time. Yeah. So that establishes that MYC maintained cells do retain pluripotency. Now, we wanted to sort of attain some more evidence that MYC was downstream of STAT3. And one of the tricks we've used is that if you express a mutant form of STAT3, this Y705F mutant, it gets recruited to the receptor, but it doesn't get phosphorylated, so it acts as a dominant negative. And essentially what this does is to block all downstream STAT3 signaling. And under those conditions, when you express a dominant negative form of STAT3, the cells will differentiate much in the same way as the cells would if you took away LIF. Now, what we wanted to do was ask the question, well, if we express MYC, can this override dominant negative STAT3? And that's exactly what you'd predict if MYC was downstream of STAT3 and the major target of STAT3 signaling. So you could bypass all upstream signaling if you activate the main downstream target. And this experiment was done by making double transfectant cell lines. We have the MYC-ER on a puro marker, wild-type STAT3, and on the other cell lines we have MYC-ER and then dominant negative. And essentially, under conditions where cells would normally differentiate in the presence of dominant negative STAT3, activated MYC can maintain self-renewal as shown by this alkaline phosphatase activity. Okay, so this actually gets around another potential problem in that some differentiated cells can make LIF and then feed back onto the existing stem cells and help retain self-renewal. What we're doing here is just bypassing all forms of LIF-STAT3 signaling by expressing a dominant negative and then asking, can MYC maintain self-renewal? And I think this demonstrates that nicely. So we've shown that MYC can maintain self-renewal, but is it necessary for self-renewal under normal conditions? Is it normally involved in promoting self-renewal in normal ES cells? So the experiment that we did to address this was to make cell lines expressing a MYC-ER fusion, but rather than having the MYC T58A or the wild-type MYC fused to estrogen receptor, we had a mutant version which lacked the transactivation domain of MYC. And in a number of cell types, this has been shown to act as a dominant negative. So essentially, when you make this form of MYC, it will be expressed, it will dimerize with its binding partner MAX, it will bind to most of the regulatory proteins, promoters that it normally regulates, but it won't activate transcription and therefore will compete with the endogenous c-MYC and will block normal c-MYC function. And then by inducibly expressing this dominant negative, you ask the question, is this compatible with maintaining self-renewal or not? So here's the alkaline phosphatase assays. We start off with the population. We switch on the dominant negative for 6 days. We start to see a loss of alkaline phosphatase activity and a clear morphological change of the cells, which is indicative of differentiation. And then if we look at genes, the gene expression markers, OCT4 is on to start with, but if we switch on the dominant negative MYC, it disappears, as does NANOG, which are both stem cell markers, and we start to get the upregulation of differentiation markers such as brachyuria and SOX1. So by inactivating MYC function in ES cells, we can promote differentiation. So we have evidence now that MYC is both sufficient and necessary for self-renewal. So this is where we're at the moment with mass embryonic stem cells. We have LIF, which binds to the receptor, which activates STAT3. And at the beginning of this study, we didn't know what was downstream of STAT3. We can now firmly place MYC downstream of STAT3, as it's both a direct target of STAT3 transcriptional regulation, and we've shown that it's sufficient and necessary to maintain self-renewal. In a number of tumour systems, it's quite obvious that STAT3 just doesn't activate c-MYC. It works in collaboration with other genes. MYC has a known pro-apoptotic activity, and what you do tend to see when you express high levels of MYC in ES cells, you do get some apoptosis. So we think that there are other target genes which are involved in suppressing apoptosis, and the PIM1, PIM2 kinases, we think, STAT3 kinases which suppress the c-MYC pro-apoptotic function. So here's the sort of the model at the moment. We have LIF feeding into STAT3, and STAT3 works principally through the transcriptional regulation of c-MYC. Now, under normal conditions, when self-renewal is promoted, you have low levels of GSK3 beta activity. Now, we don't know what suppresses GSK3 beta. It could be Wnt, because it has a known role in suppressing GSK3 beta. It could be LIF. LIF is known to promote PI3 kinase-dependent signaling, or it could be factors in serum, such as IGFs or insulin, which also can be involved in suppressing GSK3 activity. However, when you remove LIF or Wnt, the roadblock on GSK3 suppression is lifted, and then this then allows for MYC to be phosphorylated on T58. It becomes ubiquitinated, and then presumably you lose self-renewal. And we've shown if you lose MYC function in ES cells, cells differentiate. Now, in terms of how Wnt and LIF fit together, there's a drug known as Bio, and in fact, many GSK3 beta inhibitors will do this. It's been reported that GSK3 beta inhibitors will maintain human and mouse embryonic stem cell self-renewal, and we think we have a mechanism as to how this works. We think it works if you're talking about Wnt-dependent signaling. It could work through suppression of GSK3, which regulates c-MYC transcription through beta-catenin, or it could work through suppression of GSK3, which is involved in phosphorylation of T58. So the two main levels of control with regard to self-renewal, which we've identified as being important with regard to MYC dependent self-renewal, a STAT3 dependent or Wnt dependent transcriptional activation, and also the suppression of GSK3 beta activity, which allows for stable form of MYC to persist. So I guess there are two questions which sort of persist after this study. How is MYC actually imposing self-renewal and maintaining the pluripotent state? So what is MYC working on? And we're sort of trying to do some transcriptional profiling, some biochemical analysis in terms of what MYC is partnering with to block differentiation. Of course, MYC has well-defined roles in driving cell proliferation. So obviously we think there are links between c-MYC and the cell cycle studies that I began talking about. And the other issue is, well, we know that STAT3 and LIF are not important for self-renewal of human cells. So is there any overlap between the self-renewal of human embryonic stem cells and mouse embryonic stem cells? And I think that's an issue that we're interested in addressing. People who did the work, Peter Cartwright, Cameron McLean did most of the MYC work. Elaine Stead, Renata Fast, Joe White, and Dwayne Rivett contributed to the cell cycle work. Thanks.
Thanks, Steve. Let's take a few minutes for questions. John McDonald?
So you showed that overexpression of MYC is necessary and sufficient for maintenance of pluripotent state. But what happens when a cell goes on to differentiation? As far as I know, if you overexpress MYC, you do not revert back to an embryonic stem cell. So what else is going on?
Yeah, I just want to clarify something before I answer your question. The levels of MYC that we're expressing from a transgene, in fact, are equivalent to what you'd normally get in the ES cell anyway. So this is not an overexpression artifact. You have high levels of MYC in the stem cell, and we're expressing equivalent or actually lower levels. So in terms of reversibility, well, there are other things required for self-renewal as opposed to MYC, and we focused on the LIF Wnt pathway, what could be downstream of LIF and Wnt. There are going to be other factors involved in self-renewal that we haven't identified yet. And clearly switching on MYC is not sufficient to restore a more primitive pluripotent state. So I guess what you're sort of suggesting is, well, if you express MYC in a differentiated cell, can it de-differentiate? And the answer is, well, not to my knowledge, but it could be part of the mechanism that's involved in de-differentiation in addition to other things. So it would actually be interesting to look to see what happens to MYC when cells in de-differentiation systems.
You know, not being a biologist, I would have thought that as you differentiated, your cell cycle time would get longer and longer, and yet it looked, if I understood the one early graph, that the EPL cells actually had a shorter cell cycle time. So that
Yeah,
was
there's two key changes in the proliferation in the embryo and in vitro. You have this cell cycle time of around about 14 hours, which is fairly fast. Then it accelerates down to about 4-5 hours, and then it slows down. So you sort of accelerate and then decelerate as part of a developmental program. So what you saw with the ES cells and the EPL cells represent the very fast, most fastly dividing cells in the embryo. They're like the 5-hour cells from the embryo.
Understand why there might be that type of
It's a great question, and you know, why do you need to divide quickly? Well, you know, it could be due to developmental timelines, you know, but and another obvious question is, well, do human pluripotent cells in an embryonic context divide as quickly? And I don't know the answer to that question. There's a great deficiency of human embryologists who we can interact with, and it would be really good if we could go and talk to someone who could say, yeah, this is the developmental profile in a human versus a mouse. But I really don't know how the proliferation of pluripotent cells in the human embryo goes.
Yes. So Steve, you talked a little bit about how you thought MIC might be playing some global role in chromatin. But then at the same time you talked about how specific downstream pathways were carried out downstream of MIC. And then you said something about how these individual pathways, you know, would be carried out individually given that MIC was present. So were you implying that MIC was just potentiating a lot of different genes and then the different signals were then executing certain ones, or that MIC was actually changing where it was remodeling things?
I think let's just take the stem cell situation. What I think MIC is doing, it's binding to perhaps hundreds of genes. Could be 100, it could be 200. But what I think it's doing, it's marking genes required for a stable genetic state.
In a sequence-specific manner or in a MIC-specific?
Well, it binds to promoters in a sequence-specific manner, I think, if that's what you mean. So what I think is happening in the stem cell, it's binding to a certain set of genes that are required for a stable genetic state, which establishes cell type identity. In another context, it'll bind to different sets of genes which establish another cell identity. So MIC is not just involved in the differentiation or controlling self-renewal of ES cells. It's been implicated in other sort of differentiation paradigms. But it's a very, I mean, all I can say is, you know, 10% of genes are marked with MIC, and it's an incredibly large number. And that just makes it a very difficult problem to solve.
Other questions out there? Well, I've got more, but I'll hold mine. Steve, thank you very much. That was a great tour for us.
Thank you.